Hi there, this is a video about genetics and Punnett squares. I haven't done a whole lot of biology videos, but I had a request for this, so we'll give it a shot. So that's what we're going to talk about tonight. All right, so first thing I'm going to start with a handy dandy picture of a chromosome. Okay, and I drew it as a pair there because that felt like the way to draw it tonight. So in this pair, let's say there's going to be some genes on there. So a gene is a strand of DNA, so part of a chromosome. And however you look at it, it's going to have the information for a trait. So you're going to have a gene, you might have a gene or multiple genes for hair color or eye color. Okay. You have genes that might work for diseases. Huntington's disease, cystic fibrosis. Drawing a blank right now. You definitely have a gene for blood types. And there's going to be genes for all of your hormones. So there's a gene that says this is what insulin looks like. This is what testosterone looks like. This is the formula for oxytocin, and all your different types of cells and enzymes and pretty much anything. Basically, when you talk about coding for a trait, the trait is what you're seeing, but it's just the information. It's code for a protein. Because almost everything in your body is made up of proteins. Okay, so a gene is a chunk of DNA that provides the information for something, for some trait. Now, all of the genes are not alike. So for a specific gene, it may have several versions. When we first started talking about it, to keep everything simple, we always just said that there was like, you could have blonde hair or you could have brown hair. You could have blue eyes, you could have brown eyes. Okay. Now, different versions are called alleles. So if we talk about hair, I'm just going to say it now. Sorry about the shadows. I'm doing this at night with not the greatest lighting. Hair color. Okay, so we've got blonde, brown, red, and black. Oh, I don't even want to try and list all the different eye colors. Okay. Something simpler, freckles. Do you have freckles or no freckles? Dimples, same thing. Or no dimples. Okay. So there's a bunch of them. So these things are all different alleles. Because I brought it up here, blood type specifically, in humans, there's 3 alleles. So there's one called IA, which produces A-type protein. There's IB, produces B-type protein. And that protein's on the surface of the red blood cells. And there's little I, so that produces no protein on the surface of, whoopsie, sorry, on the surface of your blood cells. So this one specifically is one that's said to have be a multiple MULTI. Oh, I hope that's, I'm having a moment, guys. Multiple alleles. Okay, so in hair color here, where we've got 1, 2, 3, 4 different choices. It's nothing fantastic. It just means that there's more than the expected 2. Okay, so that's alleles. Got to get all this vocabulary stuff straight. Oh, I just underlined what a gene was. Okay. Now, some of these genes and stuff that we're talking about, they are thousands of bases long. So thousands of pairs of A's and T's and G's and C's. Okay, so we just get lazy and we use symbols to represent them. So let's say we were talking about freckles. Okay, now, again, you can have freckles, you can not have freckles. But freckles is a dominant trait. And I know that because truthfully, it's just, I remember it. Okay, so we said you can have freckles or no freckles. So what scientists do to make life a little simpler is we use F, and that represents the gene that codes for freckles. And to make our life easy, little f, lowercase f, codes for the gene for no freckles. Oopsies. I'm still pretty excited about my erasable pens that my students told me about. You know who you are, the one that told me, and I appreciate this. Okay, so ta-da. So 2 alleles. So capital F is the dominant one, little f is recessive. So 2 different alleles. One is dominant, one's recessive. Dominant, if you remember, means it's the one can hide the other one. So someone who has a recessive trait, it can be masked by that dominant one. Hold on, we'll get there. Okay, but freckles is, this is the trait we're talking about. Okay, and these are the 2 alleles. I'm going to put that. This is the 2 different versions of it. Okay, if we talked about dimples, then we could use capital D for dimples and little d for no dimples. It doesn't matter. Okay, whatever the trait is, the dominant one gets the capital letter, the recessive gets the lowercase of the same one. And we'll put a little star by that because there is times where that doesn't happen, but 95% of the time, you're going to be able to get away with that. Okay, so alleles. So what does this mean? Well, two more words. Phenotype and genotype. So phenotype is what you see. And the genotype is basically the 2-letter code. That makes that phenotype possible. Okay? You just got to learn the vocabulary. Don't get too worried about it, but you got to learn it. So if I'm looking at somebody, okay, either they have freckles or they don't. That's the phenotype. That's it. There's no half freckles or sort of freckles. Either you have them or you don't. Even if you have one, that's considered to have freckles. Now, the genotype, well, if we have these two to work with, capital F and little f, we only have 3 options. We can have capital F, we can have capital F, little f, or we can have 2 little f's, 2 lowercase f. Now, this is easy. This is the code for the recessive trait, no freckles. So the only way we can have no freckles is with 2 lowercase f's. This one, you can see that makes sense. It's going to produce freckles because it's got a capital F there. So freckles can have capital F, capital F. It's this one that messes up people. Okay, but the moment there's a capital F there, this one is hidden. It's still going to be the same. So there's 2 different, two different genotypes for freckles. There's only one for no freckle because it's a recessive trait. So this one basically can just hide for a generation or something. Okay. Now let's try that again with another trait. Yeah, let's look at dimples. Okay, so that's the trait. And again, I'm going to tell you it's a dominant one. So we're going to use the genes we're going to use, or the symbols we're going to use to represent that trait. Now, I'm writing a lot less this time. D is for dimples. Little d is no dimples, because this is a dominant and this is a recessive. All right. These words that we use, those are our phenotypes. That's what the organism is going to look like. And let's do some math, figure out first how many different things we can get here. So we got 2 choices. So I can have capital D, capital D, capital D, and little d, little d. That's it. That's the only arrangement we can have of those 2, no matter what you do. There's no other. Oh, I should say, just in case anybody's wondering, this little d, little d are exactly the same thing. They're not like the genes line up for us. And it's just easier to always write the dominant one first. So these are, I'm just trying to figure out how to organize this pretty. We'll make it the same as last time. So when you look at somebody, either they have dimples or they don't. There's our recessive trait. That's the only way to get no dimples. And capital D, as long as there's a capital D there, you're going to have dimples. So all I need is that much. The moment I see one capital D, I know that person is going to have dimples. And this person who's purebred dimples, that's what it'd be called, or homozygous dimples, is going to look the same as that one. You can't tell by staring at somebody whether or not they're purebred or hybrid, homozygous or heterozygous. All right. One more. Let's talk about something else. Trolls, something completely useless. Because then you can't argue with what I say. Trolls can be tall or short. Tall is the dominant trait. All right, so what I want you to do is set it up just like I was doing over here. I want you to name the genes, name the alleles like this. Tell me what the phenotypes are and give me the matching phenotypes. I should have written that here. So now's the time where you want to pause the video, do the work, and then turn it back on. Because if all you do is, it's pretty passive if you just sit there and just watch me do this. You need to do the work. It's just like you could, you know, go and watch somebody play hockey or train for hockey, but if you don't actually do it yourself and practice, it's not going to stick. Okay, so pause the video. All right, I'm going to trust that you did it. So what doesn't really matter what letter you chose, but I tend to make my life easy. And if I'm talking about tall and short, and tall is the dominant one, I'm going to use T. So I have to think later on, like, well, what does B stand for? Well, just go with T. So T is for tall, and little t is for short. You're not allowed to use two different letters yet. That comes later. Okay, so our phenotypes. Well, my trolls can be either tall or short. And the tall, well, actually, I'm going to do that over here, what I've been doing. Let's figure out those three pairs. There it is. That's it. It's only three options here. So tall can be capital T, or capital T, lowercase t, and two lowercase t. I'm hoping the alleles is making sense. Okay, so these are different alleles, and these are phenotypes, and again, the letters are genotypes. When we're talking about genotypes, there's a couple more words. Maybe I should throw them in now. One of them is homozygous or purebred. And they mean exactly the same thing. And I don't know, I always think it depends on who you're talking to. If you're talking to somebody in biology class, they're probably going to say homozygous. If you're talking to somebody who raises dogs or cattle or, I don't know, chickens for a living, they might be talking about purebred. Either way, what it means is the two alleles are the same. So we talk about trolls, capital T, or little t, little t. Okay, so this will be called homozygous. Dominant. And this one be homozygous Recessive. And maybe it will help you if you think about the word purebred is it's pure breeding. So if I spend money and go buy a purebred, well, let's say lab, my jelly bean, who you may or may not be able to hear in the background, she's chewing on a chunk of rawhide. She's rather loud, but she's keeping me company tonight. I got a purebred lab. Okay. I bet you can all imagine what she looks like. She's black, she's kind of goofy, and she's right now she's about 70 pounds. So she's a fairly big dog. Cool, purebred. Okay. Or if somebody bought a Rottweiler or a Chihuahua, you guys all know what those look like because they're purebreds, right? You kind of know what you're going to get. You even know a little bit about their personality. Like labs are going to love to retrieve. I don't know. Chihuahuas are yippy. I don't know much about them. Okay, but it's pure breeding. If I just tell you I got a dog, well, that opens up a whole lot of things. The first thing you're going to say, like, what kind of dog? What does it look like? But a purebred, if I say I got a lab, you know what I'm talking about. The opposite of this, or the alternative, is heterozygous. Ugh, making a mess. Sorry. Oh, still love my pen. Or hybrid. And that means the two alleles are not the same. So if I use my troll example, it's capital T, little t. And you don't have to say heterozygous dominant. No, it's just heterozygous. The moment you see heterozygous, it's one of each. Okay. And honestly, I just think of it as like heterozygous. Hetero means different, just like heterosexual. Heterosexual is a guy and a girl, whereas homosexual is two guys or two girls. They're not the same. Okay. And again, heterozygous is more sciencey. This might be something that you might be talking to a farmer. You might grow hybrid tomatoes, and a hybrid tomato might have strong stalks from one parent and lots of tomatoes from another parent. And when you do the cross, you get both of those traits. A farmer might, I don't know, cross, I don't know enough about the breeds, but two different breeds of cattle. And maybe you've got one that's a really good mom, but she's not good at calving. She always needs help with calving. They might cross that with another breed of cattle that aren't the greatest mom, but they breed really easily and they're able to, their calves don't need, they can have the calves without assistance. Okay, so you get a nice hybrid there with the two traits going on. Okay, so homozygous, heterozygous. Are we there? Any questions? Okay. So that's kind of the introduction. I guess we should get on to Punnett squares now. How long is my video here? I think we can do some Punnett squares. No, maybe I'll do the practice sheet later. Okay. I didn't really plan this. It's just on the fly. Oops. If you could see. Okay, so Punnett squares are just a way to organize when you cross two organisms. It's the simplest way to make sure that you figure out all the possible offspring. I think that's the easiest way to explain it. Okay, and they are always going to look, okay, I draw them like this. You may say, oh, I didn't draw that right. Let's go longer. Okay, they're going to look like that. And oh, here is where the gametes are. Okay, so sperm and eggs. And on the inside This is where the offspring are. So I think I want to do just some crosses first, and then we'll do a word problem where it makes sense. So for now, oops, I need to go longer. All right. And it's just like multiplication. Okay, so this t and this t, so these two go together in this box. So this t comes down, and you know what, I could even say this t comes down. So this one comes straight down like that. And these guys are going to go over. So this is going to go like this. This is going to go like this. This guy's going to come over like this, like this. Ta-da! Sorry, it's all messy over there. That's it. That's all there is to a Punnett square. Okay, your turn. Take a minute, draw my delightful Punnett square, draw the same one, and fill it in. Pause the camera, pause the video. All right. So I'm going to try it that way. So this one just comes down. So everything in this column has that letter. Everything in this column is going to have little t. Now if we go this way, I don't know if you caught that. I kind of cheated because I always like to put the capital letter first. So when we did this Punnett square, it really wasn't very interesting because every single one of those babies turned out to be capital T, little t. Each one of these is heterozygous. But over here, oh, look at the variety. This is kind of like taking these two and breeding them. Capital T, lower t. We got a purebred, and another purebred, and a couple of hybrids. So you can get all kinds of variation. The nice thing is in biology 30, you only have to worry about one trait at a time. So your Punnett squares are only going to be this size. Take university, biology, and they're going to get bigger, and it's way more fun. I think, I don't know, but I know we used to do them with dihybrids. So it was like 16 boxes. And once I made my students do one that had 64 boxes, but then I had to mark it and then, well, let me say never again. All right, let's have a question. Then we're going to talk about trolls because that seems to be a good one. So a purebred tall troll, right? Because if I just stopped at purebred, you're like, oh, purebred tall, purebred short. You got to know. Has babies. With heterozygous. Oopsies, that's not a spell troll. Heterozygous troll. Period. What are the possible offspring? Okay, so first thing you got to do, and I may change my numbering here, but I'm going to think first thing I want to do is define whatever, alleles. Now, this question in itself actually doesn't tell me enough. But from the 5 minutes ago in the video, I said tall is a dominant trait. So T is for tall, lowercase t is for short. Because that way, whatever letters you're using, I'm understanding. right? So if you decided to use B for tall and little b for short, I'm like, okay, I know what you're doing. So second thing you want to do is you want to determine the genotypes of the parents. All right, so we have one that's purebred tall. So I'm just going to write that over here. And it has babies with a heterozygous. All right, so purebred tall, that's easy. All right, it's got the tall trait, capital T, that's purebred, had to be the same. Heterozygous, I don't even have to think, one of each. And by the way, what would this troll be like? Would it be short or tall or medium? Okay, medium's not an option yet. It would be tall because it has that capital T. So we got the genotypes of the parents. Now I think step 3 would be draw the Punnett square. Oh, I'm sorry, I keep moving stuff. All right, now this is the part where people get messed up. Okay, let's say that this is the dad. That's the daddy troll. So daddy troll is going to make sperm. When sperm form, these two letters, they're going to separate, right? Then it goes a process of meiosis. So we're going to get each sperm can be carrying, well, it's not too exciting, a capital T. So we need, what's the word I want to use? Segregation of the chromosomes. I think I'm just going to write create gametes. But in the back, I'm going to say the alleles split. So on the outside of the box, you are only allowed to have single letters. So those are my two sperm. So here's Mama Troll, and she is heterozygous. So she can have a capital T or a little t. Create gametes, allele split along the outside of the square. So step number 5 is fill in the square. This is fertilization. This is making some babies. So I'm going to do it the way I normally do. So this T and this T go there. This T and this T go there. Here we have a capital T and a little t. And we have the same thing over here. Okay, and I guess question 6 is answer any questions. I didn't really do it on purpose, but you know what? What are the possible offspring is pretty darn vague. Usually it'll say, what are the genotypes of the offspring? What are the phenotypes of the offspring? How many of each you got? I was pretty vague there. So let's cover all our bases. So what are the possible offspring? Okay, so if we want to talk about genotypes, there's capital T. So that would be homozygous dominant. And we also have the heterozygous. Okay, because remember, these are the babies in the center. That's the only choices we have. Yeah, as far as I'm gonna go. And the phenotype of the offspring, well, what do they all look like? Well, you know what? This one's tall. So is that one. So everybody's tall. All the babies are tall. Ta-da! And that, my friends, is how you do a Punnett square. And I don't want to make this video too long, so I'm going to stop it there. And happy genetics.